Hi, my name is Barbara McPherson, and my presentation is over G protein coupled receptors and how they function in cell communication. In this presentation, I will discuss cell signaling, what it is, the components involved, the various types involved. I will also explain what each component is and the steps involved in the signaling process. I will discuss what GPCR is and the mechanism of how it functions in cell communication. Cell signaling is a process of communication between cells in the body. This involves signals being sent out by molecules and received by target cells. Cell signaling involves a process known as signal transduction, which according to Biology Dictionary is the process of transferring a signal across or through a cell with the help of receptor proteins. Signal transduction is where one type of signal is converted into another. For example, a target cell converts an extracellular signal into an intracellular signal using various pathways. Cell receptors convey messages from internal sources via intracellular signaling using these specific pathways. And the image here just shows a little example of cell signaling, where you have a cell here, and then you have a ligand or a molecule, then you have a receptor. This is the cell, the circle part. This ligand is going to bind onto this receptor, come inside the cell, and do some communication there, and then get a response out of the cell. We'll discuss that in more detail further down the presentation. Signals can vary in their range. They can be short or long depending on what type of signal is being sent. There are four types of chemical signals. There's autocrine, direct, endocrine, and paracrine. Now, autocrine is where a cell targets itself. And this is usually seen in early fetal development to ensure that tissue development is correct. Direct is where the cell targets other cells. This means that the cell uses signals across gap junctions. These junctions are connectors between the plasma membrane of neighboring cells. Small signaling molecules can diffuse between these cells. That would be the second little picture right here. The first one was the autocrine. The second one is the direct using gap junctions. The fourth one here is endocrine. Endocrine is where hormones, which are signaling molecules, are released into the bloodstream. They're picked up by target cells somewhere in the body. This is a long distance type of cell signaling. The next one is called paracrine. It's where cells target nearby neighboring cells. This type of signal moves fast. An example of this would be signals across synapses and nerve cells. It happens very quickly, and it happens through diffusion. And then this image shows you the types of cell signaling and how they work. The autocrine, it just signals on itself. The direct is between gap junctions. Paracrine is between neighboring cells, and then endocrine is between long distance. You see here that on the last one here that the hormone between these two target cells, that you got a bloodstream here, so it's going to be long distance. Okay, so ligands are various types of molecules. They're considered signaling molecules that will bind to proteins, or receptors or receiving molecules. They can be individual ions or atoms, or they can be large complex molecules made from many atoms. Some examples of ligands include oxygen, dopamine, insulin, or acetylcholine. As you can see from this image, a ligand is like a key and will only fit in a particular receptor. So in this image here, you see this ligand in red right here is the ligand. And it wants to try to fit into this receptor on this cell. But it won't fit in there because the shape is wrong. So it has to come over here to this next cell. And this receptor that's shaped like a Y will allow the fit of this ligand. So they're kind of like a lock and key, kind of like the enzymes or the lock and key mechanism, that the ligand will only fit a particular receptor if it, it'll only go in a particular receptor that fits. So let's see. Receptors are receiving molecules that bind to ligands on the outside of the cell, as the picture shows. That's hence the name cell surface receptors. This binding causes a signal transduction, which converts extracellular signals into intracellular signals because of a change in the shape of the protein. So cell communication with cell surface receptors involve the application of incoming signals using enzymes or ions that cause a cascade effect by acting upon one another. This cascade effect is what causes the target cell to either switch on or off, thus forcing a physiological change in the cell. And that's what this picture is showing you here. It's showing you the ligands and the receptors. Next, we'll discuss cell surface receptors. So cell surface receptors, there are 3 cell surface receptors. There is ion channel coupled receptors. This is where they allow for the passage of ions such as sodium or potassium, calcium, chloride, those type of ions. They allow passage through the membrane. The ion channel's main function is to regulate cellular excitability. There are 3 types of ion channels. There's voltage-gated ion channels, which open and close in response to membrane potentials, such as action potentials in nerves. There's ligand-gated, which open when a chemical message is received. That would be like a neurotransmitter. And then there's mechanically gated, which opens in response to a physical deformation, such as pressure or touch. The second type of cell surface receptors are G protein coupled receptors. They are coupled to G proteins inside the cell. Now, they constitute the largest family of cell surface receptors, with over 700 GPCRs in humans. The GCPRs act on a variety of extracellular signaling molecules, such as hormones or local mediators or neurotransmitters. The molecules that bind GPCRs are either proteins, small peptides, or amino acids or fatty acid derivatives. Each one of these molecules work with a different receptor to perform a different desired function. The third class is enzyme-coupled receptors. These are transmembrane proteins, which means they have extracellular ligands that bind them on the extracellular side of the membrane. Enzyme-coupled receptors use enzymatic activity and are important in the role of cell apoptosis, cell differentiation, and cell division. This type of pathway consists of an intrinsic enzyme in the cytoplasm or is coupled with an enzyme. There are 2 binding sites in this type of pathway. One is on the outside of the cell, and the other is on the inside of the cell. And this picture just kind of shows you the 3 different kinds of receptor types. So recall that the signal transduction is relaying a signal across or through the cell with the aid of a receptor protein. So this process is done through a pathway where one receptor initiates another receptor, turning these receptors and enzymes on and off. So in this diagram, you can see that this first little box here, there's no ligand. So if there's no ligand to attach to the receptor, then the pathway is off. Everything on here is off. excuse me. But when you have a ligand come in here and attach to this receptor, it turns on this first cell. So the ligand activates that receptor. And then this receptor activates the protein over here. And then the protein at the membrane activates the protein in the cytosol. Because this line here indicates that this is the membrane. This is on the outside of the membrane, the top of the line, the bottom line is in the cytosol. So when that ligand binds right there, that receptor activates that next protein on the cell membrane. And then when that protein is activated and turned on the membrane, it activates the protein here in the cytosol. And then when the protein in the cytosol activates the final target here on the pathway. And then you get a response from a target cell. So that's the signal transduction pathway. We'll discuss that more when we talk about the G-coupled proteins. Okay, the steps in cell communication. There are 4 main steps involved in cell communication. Regardless of the pathway taken, these steps are similar in each pathway. So the first step is reception. Now this is where the information from the environment, a ligand, is detected by the cell surface receptor. The ligand will bind with the appropriate receptor. That's this step right here, the very first one here, where you see the signaling molecule, the ligand. Okay, the second step is transduction. Transduction is where the binding of the ligand to the receptor will initiate a series of events, thus activating the protein and causing a conformational change. So what it's going to do is when it binds right here, it's going to come inside the cell and click onto this, and it's going to activate this and activate this and activate this, and it's going to transduce all the way down the cell. Step 3 is a response. This is where a target cell will respond to the prior series of events. And then step 4 is resetting. And this is where the cell resets back to an inactive state. We will go into more details on these steps using the cell surface receptors and pathways later. Okay, so what are G-protein coupled receptors? G-protein coupled receptors are the largest family of integral membrane proteins, and cells use them to convert extracellular signals into intracellular signals. The structure of G proteins consists of a 7-membrane spanning alpha helix segment that is separated by internal and external loops, meaning that these receptors have a single polypeptide chain that weaves in and out of the cell membrane 7 times. As you can see that on this first image on this first picture on this image here, you see this is the cell membrane or the plasma membrane. Here you up at the top you have the extracellular space, and then the lighter blue you have the cytosol, and then this green thing that's winding in and out of this is the G protein coupled receptor, and you can see it spans 7 times. If you count them 1, 2, 3, 4, 5, 6, and 7. So this weaving forms this cylindrical structure that has a deep ligand binding site in the center. You can see that on the bottom picture here. This is the G protein coupled receptor here, and the one in red is the binding site deep in the center of it. Now these receptors, I told you earlier, they were the largest receptors. Now these receptors contain 5 families: rhodopsin, secretin, glutamate, adhesion, and frizzled and taste. They function in most hormone and neurotransmitter responses. These G protein coupled receptors also play a huge role in taste, vision, and olfactory and smell. So the mechanism of a GPCR. So you can't hardly see really because this picture's kind of small, but maybe I can walk you through it. So when the GPCR is not bound to a ligand, both the GPCR and the G protein are inactive. So the very first picture in this image here shows the GPCR, and it does not have a ligand to it, so it's inactive. There's no binding to it, so it's inactive. Now, this inactive G protein consists of, over here to the right of this helix, and you'll see it consists of an alpha, which is this funny looking little, looks almost like a, I don't know, like a teapot or something there, an upside down kind of teapot. But it consists of, that's called the alpha subunit. The next one is a beta subunit in blue, and the one in light green is a gamma subunit. Also, along with this little handle right here on the back, on the bottom side of the alpha subunit is called a, it's an AH, it's called a alpha ACE, let me see, I can't hardly see it very good, alpha domain, AH domain, and then this top part here is called a RAS domain. And then you also have a bound GDP molecule inside here. All of this is still in the inactive state. Okay. So when a signal molecule, a ligand, comes in and binds to the GPCR, it activates it, and it causes a conformational change in the structure of the receptor. So here you can see that it's flat right there. When the ligand binds to it, you can see it changes shape and it becomes kind of rounded on the end so that it will fit down inside this G protein. So the signal molecule comes in, it binds it to the GPCR, it activates it, causing a conformation change, which then will activate the guanine nucleotide exchange factor. And that is in here, the GEF. Now, this change allows the G protein to bind to the GEF domain. And this process is what replaces this GDP here. This GDP is going to open, this arm is going to open up, this AH domain is going to open up, and that GDP molecule is going to come out. It's going to be kicked out and replaced with the GTP molecule. And that replacement of the GTP molecule activates the G protein. And that's all done by GTPase enzymes. Now, upon activation of the alpha subunit, once it activates, the alpha subunit will dissociate from the GPRG protein. Okay, see here on the second set here, the alpha and the beta is connected to, or the beta and the gamma is connected to the alpha. Well, once that binds and that GTP activates that G protein, the two subunits, the beta and the gamma subunit, will dissociate from the A subunit. The alpha subunit dissociates from the GPCR G protein complex and binds to an effector enzyme inside the cell, which activates the enzyme. Now, the activated enzyme will generate second messengers such as CAMP, cyclic AMP, and it will just cause a downstream signaling cascade event. It's not shown on this picture. They don't show it on that picture. But the second messengers will activate various proteins, which will generate particular cellular responses. And it will continue to do that until the hydrolysis of GTP back into GDP. And that action will deactivate the enzyme, and then the cycle starts over again. And here I have just another kind of picture of it that's showing you that how this process works is by the binding of a ligand, which can either be a hormone or a neurotransmitter, to a G protein that activates the GPCR to undergo a conformational change, which then activates the guanine nucleotide exchange factor, the GEF. And then the GEF then activates the monomeric GTPase, which will replace the guanosine diphosphate, the GDP, with guanosine triphosphate, GTP, which then will activate the G protein. Once activated, the alpha subunit dissociates from the complex and binds to an effector enzyme on the cell to activate it and causes a response. So here it's showing there's no response here. But when the signal molecule binds to the G protein, then the G protein, the alpha subunit exchanges the GDP for the GTP. Then the alpha subunit here, it shows, see, they're connected here. Then here, when that happens, it shows that the alpha subunit dissociates away from the beta and the gamma subunit. And over here, the next step is that's going to be the hydrolysis of the GTP back into GDP, causes the dissociation from the effector enzyme, thus deactivating the enzyme and returning the cell back to its normal inactive state. Okay, so that was how how the receptor enzymes, how that worked, how that G protein coupled receptor worked. Just for a little bit of information, I found this kind of interesting, that although this pathway works in a whole lot of our body systems, and also it's good for like over 50% of the drug targeted cells. It's also a biological example of how this pathway works is seen in patients with cholera. Cholera is a bacterium that enters the intestinal tract of humans and causes severe dehydration and diarrhea. And how this happens is the bacteria, once it gets inside the gut, it will produce a toxin called the cholera toxin. This toxin alters the alpha subunit of the G protein in the lining of the intestines. Now, this action stimulates the enzyme adenyl cyclase, which is going to be one of your second messengers. While the G protein is in the active state, more and more of this adenyl cyclase is stimulated, causing a deluge of chloride ions and water into the gut. And it's this ions and all this water that causes the diarrhea, and the diarrhea is what causes the dehydration when you get cholera. And so I just kind of thought that was a neat deal to put on there, even though there's lots of biological examples I could have used in these pathways. I just thought that one was kind of neat. So, and that's the end of my presentation, and there lists my references, and I thank you so very much for watching, and have a good day.